Hi, in this guide, we are going to discuss how to do the clonotyping analysis and extract TCR and BCR clonotypes from raw sequencing data. In the previous guide, we already considered and learned how to define the project, how to import samples, how to import raw sequencing data and metadata into our project. And here, for the purpose of this guide, I have already a preset up project with bulk data. This is a paired in Illumina data. For each sample, we have two files, R1 and R2, some metadata. And this is about TCR BCR sequencing. So let's do the clonotyping. To do the clonotyping, the next step, once we have the project set up, is to add the block for clonotyping analysis. And there are a couple of blocks for clonotyping, specifically one that we are considering here as MiXCR clonotyping. We have other blocks like MiXCR amplicon alignment for scaffolded artificial sequences, or let's say a CFE for a special type of libraries, which are single chain variable fragments, etc. But for the standard bulk or single cell TCR BCR analysis, we are using MiXCR clonotyping, which runs under the hood the standard MiXCR. So we just added this block, and we have settings here on the right. The number one setting that we should specify is the data set that we are going to analyze. And here in samples and data, we have only one data set added, so it's RNA-AR data. So I'm selecting this data set. And now there are two options: to either use a built-in preset or preset from file. And for built-in preset, we have a list of presets, and each preset is actually correspond to some commercially available library preparation kit, like for example, 10x Genomics, yeah, or let's say Illumina AmpliSec, etc. And those presets are actually in one-to-one correspondence with the presets from MiXCR website. So if you go to the MiXCR.com, let me just show you MiXCR.com. Yeah, in the docs, in the built-in presets, there is a list of presets. There is a list of, sorry, let me make it light. There is a list of presets that you see here on the left. And actually, it's very, very convenient because for every preset, for every library preparation kit, you actually can check the structure of the library, which is very nice. So here, returning to the presets, if we have a commercial kit used to prepare the data, then we are selecting it. Let's say this is a single cell data. Just for example, this is not single cell data, but in case if it is 10x genomics chemistry, then we have additional options like species that we need to select. Yeah, and the MiXCR reference library supports various species and the receptors. In case of single cell, you need to specify which specific type of cells was used here. Yeah, it might be just TCR alpha beta, or in some single cell protocols, it might be multiple of them. Yeah, and that's it. That's our parameters. In the case of this data that I have for this guide, the library preparation protocol is actually selected driver map adaptive immune receptor analysis for human species. And a nice feature of this kit is that it allows to profile simultaneously all 7 immunological chains. Although this is a bulk sequencing and each chain is amplified separately, but they can be analyzed with this chemistry simultaneously. So I am selecting all of the receptors, or that's equivalent to selecting all of the chains one by one here. So with this setting set up, I already can click run button, but I'm not doing this. This is my strong recommendation. Let's open advanced settings. And the first parameter here is to take only this number of reads into analysis. And this is a really important thing because with this option, you can specify, let's say 1000. Take only 1000 of reads into the analysis from each sample. And let's click run. This actually, let me return to settings. This actually a very convenient thing, allowing you to do like a dry run of the analysis by picking just a few reads from every sample. The whole analysis on the whole data may take like literally many hours or sometimes days for a big project. But with this option, you will have the analysis complete really fast, and you can build the whole downstream pipeline of your project. While it is running here for 1000 of reads on each sample, let's explore other parameters available here. So by default, MiXCR has and uses its built-in library of gene segments. And actually, this library is open source, and you can visit this library on the vdj.online portal under the gene library tab. This is like for all species. You can explore and download the gene, the germline base that we have. Very convenient. It's much better than NDD, actually, but for various reasons. But if you want to use a custom library, then you can provide this library as a file. And the format for this file is very standard. For the NGT, you can actually download it from MiXCR website, or this is from file option, or we have also, if you have a custom species or just a custom artificial genes or whatever, yeah, or you use with a model species like humanized mice, etc., then we actually have a dedicated block. It's called library, MiXCR library builder, which allows you to add faster sequences for V, D, G, C, and optionally C genes of different logical chains, assemble this as a library, and then use this library. I'm not doing this because we just have human, yeah, and then use this library here under the custom library option. So that's about the reference library. And the other two parameters that we have here is set memory per every sample process and set CPU number per every sample process. Those are actually all those parameters are optional, yeah. But these two, memory and CPU, control how many resources are allocated for each run of MiXCR for every sample. And you may play with these parameters to increase the level of parallelization on your backend. So if your Platforma is running on the server and you have a lot of CPU and memory, you can play with these parameters to allocate multiple MiXCR processes simultaneously. For example, if you have 64 gigabytes of memory and let's say 32 CPU, then if you specify 16 gigabytes per process and 8 CPU per process, then 4 analyses will be running simultaneously. So, but let's return here. And when you click Run button, you will see the progress of the analysis for each sample. Yeah, and here we already have some samples processed. And while it is running, you can click, or when it's already complete, you can click on the individual sample. Let's say I'm clicking this one to explore in more details the quality controls and quality reports for this sample. Actually, you already see a nice overview of the quality checks. And this is an important thing. I didn't mention, but with MiXCR clonotyping, you don't need to run FASTQ for QC of your data. It's all done by MiXCR automatically, providing you a really rigorous overview of quality checks. Like here, we have the proportion between the successfully aligned reads and reads that are not aligned at all, and with the reasons why they are not aligned. As I mentioned, for this specific library preparation protocol, all 7 chains are profiled simultaneously. So we have the distribution between the immunological chains in the data, well, nicely even. And then we can go to the quality checks. And this is really important thing and a cool feature of MiXCR. It provides you a comprehensive list of quality checks that you can use to understand whether everything is good with your library and what are the potential things that you can improve in the web lab to make it better. So this sample is good. We have only one warning that the number of reads which is used in clonotypes is 73%, which is considered to be actually low, but it's fine because we are taking only 1,000 of reads for our dry run here. So in the dry run, obviously the first reads from the FASTQ files, they always have low quality, etc., etc. So this is something expected, and it is fine. But then in other cases, when we have like alerts, let's say for example, where do we have anywhere? Well, no alerts here. But if we have alerts, then you can read what's the potential reason of this thing. And actually, on the MiXCR website, there is a section, MiXCR Quality Controls, a comprehensive section with like list of all quality checks with a deep dive explanation of what they mean, how to understand the quality issues, how to improve your library preparation process, and what's specific, maybe specific for bulk data, for single cell data, for just non-targeted data like RNA-seq, etc. So this is very, very convenient section. And here you can read a lot about the quality of the repertoire sequencing libraries. But returning here, yeah, the next step is more advanced. This is the log of the just bare command line MyCR execution. So this is what you see when you run MyCR in the command line. Yeah, this log, and these are the comprehensive report, just the raw reports which are generated by MyXCR tool under the hood. So let me close it. And while this is processing, what is our next step? So we have the clonotypes produced by MiXCR clonotyping. I forgot to mention, but we also have a QC report table here, which will be populated with the QC overview once the analysis is complete. And what's our next step? The next step is to explore the clonotypes. There are several ways how you can do this and start building your downstream analysis. The most natural one step after the clonotyping complete is to just look on the clonotypes, which can be done with the clonotype browser block. So you add this block, let me add it. So we have clonotype browser, and in the clonotype browser now, in the settings, we can select the dataset. And as you see, the dataset here is just the list of immunological chains for the data. And this is very important because in our clonotyping analysis, which by the way already completed, that's why we choose 1000 of reads, we selected all 7 chains. And since this is a bulk data, every chain is amplified separately. We can look on the repertoire for only one chain at a time, obviously. And let's say we select TCR beta, and now we have all of our clonotypes loaded. Well, for every sample here, so we can switch between samples, or in the overlap, we can see like the total, all of the clones that we have from all of the samples in a one table. Actually, we can sort by the number of samples to see that some of the clonotypes belong to many samples. Yeah, obviously the number is low because we profiled only 1000 of each for every sample, but I've here the whole project. And for TCR beta, for example, we have in total 3 millions of clonotypes. So returning here, actually Clonotype browser is a very powerful instrument allowing to, well, show different columns. Here, well, this is can be ignored. A powerful tool allowing to explore your repertoire in many, many, many ways, show different columns, filter the data, annotate your clone types, and do a lot of things. But we are not going to discuss it. We'll have a dedicated text for it. So now let me return to the other types, to the other option that we have here, the preset from file. So I have another project with VHH data of alpacas where the library preparation protocol is custom. So the same thing, we are adding MiXCR clonotyping block. We are selecting the data set, and now we are choosing the preset from file. And this is just a YAML file that you can prepare with the parameters of MiXCR analysis that match the library structure that we have. Here I have an example. So actually I have a file for alpacas, preset alpaca, and here I have this file showing you what's the content of this file. So on the MiXCR website, there are instructions how to prepare a preset YAML, and for all of the available library preparation kits, you can actually go to the GitHub and see how the preset is defined. So you can, with this preset, you can specify absolutely arbitrary library structure. You can specify UMIs, cell barcodes, sample barcodes, multiple UMIs, etc., etc. Whatever it has. Yeah, so it's a comprehensive thing, and one way you read the documentation and you assemble this preset yourself, or you can actually always ask help with just this help button, very, very convenient, which navigates you to Platforma help center where there is a live chat. You just say hello, and our team will answer you promptly and help to prepare. Or we also have community forum, community platform mobile, where you can also ask those questions and get replies. So once we select the custom preset, we also need to select the chain. In the case of this data, only monoglobulin heavy was profiled. And then the same, we have the same advanced settings. We click the run button and the analysis will start for this data. Here, for the custom case cell, here I actually have already processed this data, and you will see the same story, the same quality checks, the overview of the QCs. And if you open and navigate to the quality checks there, you will see like the hints, the same. By the way, these quality checks are tailored to the type of the chemistry used. So this quality checks are specific for the protocol that we have, whether it has VMI, whether it does not have VMI, single cell, etc., etc. And here we have a clear alert because the number of reads that we used in the clonotypes is below 40%, meaning that the 60% of data is just garbage. This is not something that we expect, but this is like clear feedback to the wet lab, and reading the documentation, you can understand what's the reason. And here, for example, we have a QC report table with like the overview of all the things, like the number of reads in each sample, number of clonotypes in each sample, number of successfully aligned reads, and what are other things available here. So that's it. This is the guide how to run your clonotyping analysis, the upstream part of every project, VDJ project. And we consider the human and alpaca libraries, bulk sequencing. Same applies for single cell. In the next guides, we will also cover how to analyze custom libraries like a CFV or fusion protocols, how to align the amplicon libraries, which are generated from scaffold sequences, etc., etc. That's it for today. Thank you very much, and looking forward to our next guides.